Okay, so yesterday we finished off and we had a few more things to talk about with topic 19 and biotechnology and just some kind of interesting things around these technologies I wanted to share with you. First is that DNA technologies have been used over the last actually almost 20 years now looking at ancient DNA. So let me just talk about that a little bit because there's some really fascinating things. You may be familiar with this story if you've seen this little movie called Jurassic Park or some of the sequels. This whole idea about getting DNA from amber. This, it turns out, is probably not. Hold on, here's my PowerPoint is doing something funny here. Here's my Jurassic Park. So we're not there yet. It turns out that DNA probably doesn't last this long. Experts kind of argue about this. Some people say about 50,000 to 100,000 years. So there have been some people who have claimed to have extracted some fragments from dinosaur fossils. I think there's a lot of debate over that. But if you think about that time frame, 50,000 to 100,000 years, there are a lot of things and artifacts that we do have that fit that time frame, particularly the time frame around human existence. So really fascinating studies have been done on this guy. This guy was found in the Alps, so I think it was between Italy and Switzerland. And there were a couple of people hiking, and they found a body. And initially, they thought it was just, body from somebody who had died recently. And it was realized very quickly this guy had really weird sandals or moccasins or something like that, a bow and arrow, and clothing made out of animals. And this is actually the best preserved human specimen we have, or at least the oldest one. We do have older Egyptian mummies, but Egyptian mummies often, they're treated chemically and organs are removed. And so there's been quite a few studies done on this guy. And interestingly, some DNA studies. So they analyzed and they found several DNA samples. So DNA samples. So one sample was from him. They could tell what his sample was. But when they started to look at other things that he had a sample of blood and somebody else's DNA on his knife. So human DNA on his knife, interesting. They also found two other samples on arrowheads and another DNA sample on his coat. So this tells us a little bit about this man. He clearly lived somewhat of a violent life, perhaps. He had human blood, not his own, on his knife, two different samples on his arrowheads, and some on his coat. So we're not exactly sure what he was involved in. Maybe he carried a wounded comrade, but you can see the DNA samples do tell us a little bit about him and a little bit about his genome as well in terms of his origin. He's related to Germanic people, I believe, is what they had concluded. Lots of other interesting ancient DNA samples. Some people up until recently had been arguing that maybe the Black Death was, one thing or another. And something that really confirmed what we had suspected for a long time is that this organism here, Yersinia pestis, was in fact the organism that caused the Black Death. And we know this now because we've dug up several historical remains from that time and have been able to extract DNA confirming that this is the organism that caused the Black Death. So if you don't know much about the Black Death, it was really a huge, significant part of human history. Happened in Europe and Asia in the 1300s and killed, we don't know how many people. Some estimates say a quarter, some say half of the population. This was a huge, huge pandemic. We're talking about pandemics now. This was the one that really, Black Death is an earned name. So some people wonder why modern infections aren't as bad as ancient infections. And there are a few changes. I don't know if we've teased out and figured out whether these changes make a difference, or maybe people just had worse immune systems back then, or could be some other genetic reasons, but it gives us some answers. So one more other interesting story. Interesting story about King Richard. So I don't know much about King Richard. He was a king of England. And one thing I learned about King Richard is that he was the last king of England to die in battle. So a few years ago, I think this is 2015 or 16, they found his remains, believe it or not, under a parking lot. I think they had suspected his remains were there. And so the question is, was this King Richard? So you can look at kind of the usual type of tools, carbon dating. So the skeleton was about the right age. Interestingly, the skeleton had battle wounds, which fits with the picture of what we know about the history. There were also skeletal deformities. So if you're familiar with the Shakespeare play, King Richard, he's a hunchback in there. And that is actually based on the fact that he did have a slump, apparently, King Richard. But the other thing, at that point, they were like, 75, 90% sure. And so what about the DNA? So it turns out that there was a distant relative of King Richard, and she was a Canadian. And so they took a look at her DNA, and they took a look at his DNA, and the evidence does confirm that they are relatives and that this indeed was probably King Richard. So kind of very interesting and cool stuff that's being done with this old DNA. So can we do anything even weirder? I wanted to share this with you because I see this story about every 2 to 3 years in the major news sources. So notice I even showed you where I got this from, New York Times. This isn't like History Channel or some sort of sketchy reporting. This was the New York Times. This was an older article, but I like the pictures, so I thought I'd share them with you. And so we have remains of woolly mammoths, and we have hair, we have blood samples, and so we actually have the mammoth genome now. And so one of these scientists that was involved in this project said, yeah, for about $10 million, maybe we could make a mammoth. I don't know if that's true or not. This is kind of what he was proposing. So if you look at the mammoth, and the closest living relative is the African elephant, and about 400,000 places it's different. So the thought is, okay, we could go through here and we can change the relevant codons and maybe make ourselves a mammoth. Don't know whether that will work or not. Even more recently, there's other scientists, Harvard scientists. These are big scientists saying that this will be done in 2 years. So this was 2017. I don't think we have a mammoth yet. But this was because we had found this amazing specimen was found in Siberia. And it's almost a complete mammoth. It's a baby mammoth, but you can see hair on it. It's absolutely adorable. And these scientists were saying, yeah, we could extract the DNA. Maybe we could, at least swap out certain genes for cold weather and hair, and make some sort of mammoth elephant hybrid. So kind of cool, kind of interesting, kind of science fiction-y, but who knows? We'll see what happens in the next 10 years. I'm sure the Discovery Channel will have more specials on mammoth DNA in the future for sure, 100%. Okay, so what about scarier things? Here's something done almost 20 years ago. Reconstructing viruses that in this case here, they had reconstructed this virus completely from sequences found on the internet. So, technology is like this. You can do good things, you can do bad things, you can do scary things, you can do things that are kind of not good or bad or scary. Polio virus is not necessarily scary. We have vaccines for it. It's probably something that you've never experienced in your lifetime, polio. And you probably don't know anybody who's ever had polio. But what's to stop people from doing other things? And these are some of the questions that are raised around biotechnology. But I thought I'd show this to you because of two things. One, it is kind of scary. And two, the date. We have been doing this for a long, long time. So the good news is people don't seem to be making smallpox. By the way, just for the record, smallpox is probably not, using current technologies, we probably can't be. So I just want to share with you, I had mentioned that this CRISPR-Cas9 technology was really cool. And you can basically edit DNA inside cells. So I just wanted to share with you 3 projects that I've read about in the past year or two that are really kind of interesting. One is making hornless cattle. So you're probably thinking, so what, cows without horns? Well, talk to farmers. The dehorning process is difficult. It can be, there's a lot of blood involved and it can be dangerous, and the cattle horns are dangerous. So why not just get rid of the horns entirely? The meat and everything is the same. So this has been done. I don't know if they're for sale yet or not, but it's the concept has been done. Project number 2. What about making mosquitoes that cannot carry malaria? Also, this has been done apparently in the lab. It hasn't been done in the wild yet, but this is doable. Malaria infects 1 billion people worldwide and kills, I think it's something like 30 or 40 million children a year. So this is a scourge, a huge scourge. Never mind all the other diseases spread by mosquitoes. Maybe we could do something about them too. So one last project to share with you. Oh, I don't even have it on there. I was going to show you something about making cells to be HIV resistant, but I guess I must have cut that one out just to save some time. That is the end of topic 19. So I want to spend the rest of today on the final exam and go through and tell you, hopefully you have an idea of what you need to study for now, but I'm hoping this will firm it up and help you to understand what you should focus on and some of the things you should do for studying. So there's Calvin. If you're interested in some good procrastination from studying, check out Calvin and Hobbes. He's always a funny guy and good kind of cartoons for the brain. So I think I had showed you something like this before. If not, I'm sorry, I meant to show you last week. This is the breakdown of the exam. So it's going to be 80 marks. So the midterms were 35 marks each, I think. So this is about 2 midterms. The balance, I have a few more multiple choice in terms of the weight wise. So they don't take as long as like written questions. And you can see my little note here that about 70%, maybe 75% of the exam will be the later topics. And I think I'd mentioned last day that, the way I usually do this is I'll take about 2 multiple choice questions from every topic, true or false as well. And that's kind of what I do. And then after that, all of the written part of the exam is going to be from the latter part of the course. So notice this here. You're going to need to show your ID. I think I know what everyone looks like, but for final exams, I need to be a little bit more strict in terms of how I do things. And I need to have proof of everyone's ID. So if you have a Kano ID, please show your Kano ID and make sure you have it close enough to your webcam so that I can actually see it, okay? Some people like to flash it and it's blurry. Get it up close enough to the webcam. If you do not have a Kano ID, that is fine. You can use another ID like a driver's license. If you use your driver's license, just put a little piece of tape over your address. I don't really need to know where you live or anything like that. If you don't care, that's fine, whatever. Just something I don't need to have that extra information about you. If you want to hide any other contents about your eye color or height, you can hide that too if you like. So let's talk about this exam. It's going to be used in the lockdown browser like before. The only thing is that added step of your ID. Okay, so make sure you have your ID ready. I will send out some instructions at the end of the week, and I will remind you of that in the email as well. So if you're having internet problems, I think we've done this pretty decently. So the exam is next Wednesday, and as before, I will make sure that I'm on campus and in my office during the exam time. And if you do have any glitches or issues, you can phone my office phone, and we can see if we can come to a resolution. So what topics did we cover? So this was the course. We covered topics 1 to 6 and the virus topic, and then we did midterm 1. Then we covered topics 7 to 12, and then we did midterm 2. And then the last few weeks, we've been covering topics 13 to 19. So this is all on DNA and RNA and proteins and all the related steps in between. So this is where you want to spend most of your focus. I would say a good 85% of your focus you want to spend here in learning these 3 topics: replication, transcription, and translation. Those are the things that I do want you to focus on, because they're kind of the core of everything else in these topics 13 to 19. So let's kind of go through the topics. Some of them I'll kind of whiz through quickly. Others I'll spend a little bit more time on. kind of reviewing some of the things we talked about. Topic 13, we talked about some historical experiments. So 3 of the experiments we talked about were confirming that DNA was the genetic material. So the Griffith experiment and the Avery experiment were kind of like the lab experiments where we were looking to see if DNA could transform some cells. They were looking at pathogenic cells. We weren't using pathogens. The Hershey and Chase experiment, this was a virus experiment and using radioactive phosphorus and radioactive sulfur. So we also talked about the structure of DNA. So it's important to know a little bit about the chemistry of these nucleic acids. They have 3 parts: phosphate groups, they have pentose sugars, and if you are RNA, you have ribose. If you are DNA, you have deoxyribose. So this nucleotide here, this is a deoxy. So there's no oxygen there. If this was RNA, it would have a little O on it right there like that. But this is not, so I'm going to erase that. You don't know how to erase it. Never mind. I guess I'm not. I'm just going to scribble it out. And then we have our nitrogenous bases. And we have, of course, 5 nitrogenous bases we learned about. If you are in DNA, we have G, A, T, and C. If you're RNA, you use G, A, U, and C. So U for uracil, of course. We also talked about Watson and Crick and the evidence that they used to construct their famous DNA model. And I have some of it summarized here, and we're going to go through that. So definitely you can expect 1 or 2 questions on Watson and Crick on the final exam. And so Chargraff's rules, what were they? Edwin Chargraff was talking about basically proportions of the nucleotides are the same. So he found that A was equivalent to T and G was equivalent to C. Secondly, there was the charge of the phosphate groups. So there were people that were trying to make DNA strands, and they were thinking that the phosphate groups were on the inside and the bases were kind of sticking out sort of like this. And that doesn't work because all the phosphates are negative charges, and these negative charges don't want to be together. Maybe one of the most important pieces of evidence was Rosalind Franklin's X-ray image. And this put the phosphates on the outside. It gave a width of the double helix, and it also indicated a helix. So they put their models together, and they realized that just like Chargraff's rules, that adenine fits with thymine, forming perfect hydrogen bonds, and guanine fits with cytosine, forming perfect hydrogen bonds. So if you remember, this has 2 hydrogen bonds, and this group has 3 hydrogen bonds. And that was their model. So make sure to review that because, like I said, I'll definitely have at least 1 question on Watson. So one other thing about their model was that when they built it, they realized that the DNA strands are complementary and anti-parallel. So I keep coming back to this, right? It must be important. So what does this mean? Complementary, this is the base pairing. It means that A base pairs with the complementary base, which is T, and G pairs with C, which is complementary. The anti-parallel is referring to this here. So one of the strands is in the 5' to 3' direction, and the other strand is in the opposite direction. So this strand here has the 3' at this end and the 5' at that end. So this is just a chemically way to construct the 2 strands. So DNA replication, we talked about a whole bunch of different enzymes. I'm not going to fill this one in. We did do this already in the, when we did DNA replication, I filled in this chart already. But you can use it as a review and go back and take a look at those notes or look at that video to see what this is talking about. But remember that for DNA replication, there were 6 enzymes and proteins. So like I said, I'm not going to fill this in, but I will talk about them for a moment. So first of all is primase, that is adding the RNA primer. DNA polymerase is the big enzyme that's going to add in the new nucleotides. The new nucleotides are going to be complementary to the template strand. DNA ligase is what is going to seal the gap between the Okazaki fragments. I think I have a picture here of the machinery. There it is. So there's all our friends. We've got primase here, ligase there, DNA polymerase, helicase, single-stranded binding proteins, and the topoisomerase in there. So I think I have some notes on a couple of these. So what was the helicase? The helicase is there for unwinding the two strands and separating the hydrogen bonds. The single-stranded binding proteins are going to keep them single-stranded. It's going to prevent them from re-annealing together. And the topoisomerase is there to relieve the tension of the unwinding DNA. So it's just kind of preventing it from forming knots. So with regards to DNA replication, I think I told you before, for the exam, I'm going to have a question on them. I haven't decided whether it's going to be a matching or something, but asking you about the functions of these enzymes and proteins. So make sure you're prepared to do that. I've done fill in the blank, I've done matching, I've done other things. And so you should be prepared to explain or understand what all of the roles of all of these particular enzymes are at. And also pay attention to, there are differences happening at the lagging and the leading strands as well. Topic 15 was all about the genetic code, and that was kind of an introduction to the following units. But mostly it was kind of going through and defining what these things here are, right? So these three stages I keep talking about: replication, transcription, and translation. So here's my study tip. I recommend for these things that you practice writing out the steps for them. So replication, this happens, then this happens, then this happens. Take a look at my notes, take a look at the textbook. There's other resources. And what you want to do is make sure that you have the correct terminology, okay? I know when I read exams, sometimes people are talking about transcription, and so transcription, by the way, is done by RNA polymerase, right? But instead they're saying ribosome or DNA polymerase or something like that. And all this other terminology we've talked about, things like template strand, complementary, antiparallel, all of these words are important to incorporate in your answers. So it doesn't have to be sentence form on my exams. You can use point form, that's fine. But I want you to make sure you have the right terminology, that you're thorough and accurate, okay? The long answer questions, definitely at least one of the long answer questions is going to ask you to describe one of these processes. So you're going to need to be prepared to do all three, depending on which one I ask you to do. Last tip about these is there are some differences between prokaryotes and eukaryotes, things like location, or sometimes there's extra steps. If you're a eukaryote, in a eukaryote, for example, there are those transcription factors and things like that, right? In eukaryotes, there's a lot more processing around the RNA, for example. So topic 15, remember, all these steps are done in the cytoplasm in bacteria, and it's pretty much happening all at the same time. In a eukaryote, I don't know what happened here, this should say protein. In a eukaryote, some of the processes, so the DNA and the RNA production happens in the nucleus, and the ribosomes are found out in the cytoplasm, and so the translation is going to happen out in the cytoplasm, and often, in many cases, that translation is continued at the rough endoplasmic reticulum. And so we talked a little bit about that, and I have some slides coming up to remind you of how that is done as well. Topic 16 was on transcription. So transcription, like I said, remember, we've got this word here, script, right? So if you are a scribe, you're basically copying something, and that's actually what transcription is doing, but it's the same language. It's nucleotide to nucleotide. So you can see there's our DNA, and the DNA is converted to RNA, and you can see that it's almost the same. Not 100%. We're changing these T's here, and we're changing them to U's. But other than that, it is basically a match. So the other thing to think about is we have three steps. and we have these different strands we're talking about. So in this case here, it's possible in my notes I had these strands the other way around. I may have had the sense strand on the top and the template strand on the bottom. It doesn't really matter which one is on the top or which one is on the bottom. What matters is you remember that we read our DNA from 5' to 3', and the sense strand has the start codon. So look for your start codon, and that is going to help you to figure everything else out. So the sense strand is the same sequence as the messenger RNA. That's why it's called the sense strand, because it hopefully makes sense. The template strand, on the other hand, is the strand that is actually used by the enzyme. That's the strand used by the RNA polymerase. So the RNA polymerase, where it sees a C, it puts a G on its RNA, and so on. And so it's the template, meaning that's what the enzyme is reading. Sometimes it's called the antisense strand. I don't usually use that word, but the textbook does. I will try to call it the template strand, but if you want to call it the antisense strand, that's fine as well. So here's some terms. You should be familiar with these terms and a few others, but just reminding you of some terms if you might see something in there you don't recognize. So you may want to go and study them. That's why they're on this list, to remind you that definitely these terms will end up on your final exam somewhere. So you should know what they are and be prepared to answer some questions around them. Topic 17, translation. So translation, we are going from one language to another, language of nucleotides to the language of amino acids. So this is also called protein biosynthesis. And this is done at the ribosomes. Remember, our ribosome has 3 sites. It has an A site, a P site, and an E site. And so you should know what is going on there. Like I said, make sure you go through the steps of this, know what is going on in translation, transcription, and replication. And you should be prepared to write out the steps of initiation, elongation, and termination in a long answer type of question. And so you're going to be referring to, all of the components. You've got the large and small ribosomal subunits. You've got the messenger RNA. This is the messenger RNA here, by the way. You've got your transfer RNA. This is a transfer RNA here. And of course, the amino acids. There's some terms at the bottom, right? So in your answer, you'd use these terms: start codon, release factor, ribosomal subunits, transfer RNA, and so on. So translation, by the way, in my mind, is the one that maybe students get a little confused about the most. So, make sure you do spend some time on this one, because sometimes people get confused around it a little bit in terms of what is going on. I know I have some flow charts in the notes. Write your own notes and, do it again and again as part of your studying so that you understand what is going on. Textbook is a good resource. The textbook does go into it in more details. I'm not going to ding you marks for having too many details. That's fine. Just make sure that you're complete. So let's take a look at one of these problems here. Like I said, you're going to have something like this on the final exam. I have 10 marks devoted to interpreting DNA strands. Okay, so a lot of the marks are going to be a question like this, where I'm going to ask you about it and ask you about mutations and things like that. So I have some samples here we're going to go through. So let's take a look at this. Like I said, the very first thing that you want to do is find that start codon. So if you take a look, I'm looking at the top strand. I'm looking for ATG, and it is right there. Okay, so here is a hint. In my class, it will always be in the top strand. And in my class, that will always be the sense strand. That's not always the case. I'm just trying to keep it simple for everybody this semester. If you do happen to see a start codon down here, that's not the right start codon. The question will still work for you. You're just going to lose half a mark for choosing the wrong strand. But that is not a start codon. Because remember that we are reading our DNA in this direction. We're always going from 5' to 3'. So make sure that you always read 5' to 3'. So someone has a question and asking if you'll be provided with the chart. I'm assuming you mean the genetic code on the final. Yes, I will have the genetic code for you ready on the final exam. Okay, so start off with your start codon, and then we're going to do all this other stuff. So we've got a sense strand we got to find, and I just told you that the sense strand, at least in this class, I'm always going to put it on the top. So there's a hint for you. Half a mark. Antisense strand or template strand is the other one, of course. Okay, that's a good start. The next, it says give the sequence of the messenger RNA. So this TAT here, this is usually the beginning of the promoter, but it's not exactly precise where the RNA polymerase binds. It binds roughly around there. So when you write out the sequence of the messenger RNA, I want you to write the entire sequence. Don't just start at the start codon, write out the whole RNA. So remember, the messenger RNA is just like the sense strand. So that's going to be my messenger RNA. So I'm just starting right at the beginning here. Okay, I'm going all the way to the end. And I'm copying out the sense strand. And of course, what I'm doing is when I see a T in the RNA, I am putting a U. And then all the other letters I'm copying. So T, 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 A, T becomes U, 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 A, U, and so on all the way to the end where I have that CCG. Okay, so another mark for the messenger RNA. And then the next is the sequence of the protein. So there's my genetic code. So notice I'm just underlining the triplets here of the protein, and I'm going to go all the way to the end. So basically, we're just going to copy those out and take a look at the genetic code. So AUG is on here, and AUG is methionine. So you don't need to know the full name of the amino acids. 3-letter codes is fine. In fact, the genetic code I'm going to provide, I think, just has the 3-letter codes. So ACG I'm looking for. So that is right here. And that is something called asparagine, ASN, GAA. I always find this part slow. There it is, GAA is glutamic acid. And UUA, I'm not going to circle any more of these. UUA is leucine. AUC, AUC, I saw there a second ago. Okay, that's isoleucine, that's ILE, and UAA, that is our stop codon. So that is my protein sequence, okay? If you want to be even better than that, remember there's no 5' and 3' with RNA and DNA, but there's an N terminus, which is the beginning, and a C terminus, which is the end. But that's understood when you write down the sequence. You don't have to do that. I just like to do it because, hey, I'm a biochemist and I like things a certain way. So let's take a look. That's part of the question there. That's about maybe 3 or 4 marks worth of material out of 10. I'm going to be asking you some questions about mutations. So remember our mutations, there are 3 different types. And let's take a look at how we can analyze that strand with mutations. So I think this is the same sequence. I meant it to be the same anyway, right? And so each of these will be worth probably a mark. So it says, what type of mutation is caused by a deletion at position 1? So remember that this is my start codon. So the answer to that is that this is not part of the gene. So this, give me a sentence and explain that this mutation is not in the gene. So it's it could maybe affect a regulatory component of the gene or part of the promoter, but it's not going to affect the amino acid sequence. So that's why I have NA not applicable, because it's before the start codon. So what about position 2? Okay, so position 2, we would be going from an ACG to something else. Okay, we'll just call it question mark CG. So take a look at your genetic code. So your genetic code, we're looking, where's my ACG? Here's my ACG. So I'd be going from a threonine, okay? And if I change it to, let's say, a G or a C or a U, I'm getting completely different amino acids. So on the exam, I'll probably give you something more specific. I'll say what happens if you change the A to a G. Okay, let's just go with that one. So then you'd explain that we're going from, in this case here, a threonine to an alanine. And when you're changing the amino acid, that is a missense mutation. Okay, so make sure you use the correct terminology. Remember, missense, it's just kind of like mistake. Okay, it's so it's the wrong amino acid, a mistake, an error has been made. So what about a substitution at position 3? So position 3, we got A, C, and then if you take a look, no matter what that fourth nucleotide is, you're going to have a threonine, and then you're going to have a threonine. So when you go from one amino acid and you don't change that amino acid, this is called a silent mutation, meaning that it's not affecting the amino acid sequence, even if the DNA sequence is affected. So last one, it says, what happens if we have a deletion at number 4? So take a look at this. So I'm just going to put a square around all my codons leading up to that point. So in this case here, I'm going from a TTA. Okay, so that's my original codon here. So a TTA, of course, is going to be a UUA. Okay, so I'm looking up UUA, and that is leucine. But if I delete that T, that one in red, I'm going to end up with a TAA. So of course, a TAA is a UAA, and a UAA is a stop codon. So what happens when we go from a leucine to a stop codon? That is called a, well, actually that's not the word I was looking for. That's called a frame shift, but it's a frame shift leading to a nonsense mutation. So nonsense mutation is where you're introducing a premature stop codon. So nonsense, there's got the word no in there, so no go, right? It's stopping entirely. So that's going to be, I don't know, where are we at now? I said 3, maybe another 4. It's about maybe 6 or 7 marks out of 10. And I'm going to find a few other questions to ask you about the DNA. I haven't decided what they are yet, but think about all the things we've been talking about. We were talking about things like introns and exons. Maybe I would, well, I haven't decided yet. 10 marks, so you know about 7 out of 10 marks anyway for this DNA analysis question. It's possible I may break it into 2 parts. Maybe I'd have the translation transcription one part, the mutation another part, or something like that. I haven't figured that out yet, but you have that question to look forward to. Okay, so topic number 18 was on gene regulation. And I know this topic, I threw a lot of stuff at you. Okay, I was trying to just keep it focused to stuff we had talked about to some degree in class. There were a few new words. For example, this one here, probably trying to remember what that word was. The basic unit of gene expression in a bacteria is the operon. So a lot of genes in bacteria are regulated by operons. And so what is an operon? An operon is a bunch of genes. So it could be 3, it could be 5, pretty typical. They're controlled by one promoter. And within that promoter is an operator. And so the operator is a region where a regulatory component, so a protein usually, is going to bind. So if you remember, we looked at the tryptophan operon, and here is the tryptophan operon. And so let's just go through and take a look at these parts, okay? So let's take a look. Part number 1, I thought I had this animated, I guess I don't. Part number 1, this is the promoter. So remember, the promoter is the section before a gene or genes where the RNA polymerase binds. So number 2 is the operator. So the operator is where a regulatory protein is going to bind. So 3 and 4, these are start and stop codons, start and stop. Number 5, these are the proteins that are going to be made from this. Number 6 is a regulatory protein or gene, regulatory gene. It makes its own messenger RNA, it makes its own protein. So number 7 is the regulatory protein. Find when I talk, I can't spell and talk at the same time. I don't know, I used to be able to spell. And then number 8 is the RNA polymerase. So we've answered what an operon is. What are all the numbered parts? It says here, what happens in the presence or absence of tryptophan? So in the absence of tryptophan, we'll start there. That whole regulatory stuff is not important. We have RNA polymerase binding, it makes RNA. The RNA is used to make proteins. When we get tryptophan, tryptophan binds right here. And then this thing, this regulatory protein, binds at number 2, the operator, and blocks the RNA polymerase. And so the genes are not transcribed. So take a little bit of time reviewing that. I think I've decided it's probably going to be 2 multiple choice questions. I might show you a picture like this. I might ask you what an operator is. I haven't decided yet, but I think I have decided it's going to be minimum 1, probably 2 marks in the multiple choice section on this, or true and false section. Okay, so what can we talk about eukaryotes? There are a whole bunch of things we talked about, mostly just tiny little things. And we talked about little things that happen at most of the levels. So at level 1 is the DNA level. How is DNA regulated? Well, DNA is regulated by being euchromatin or heterochromatin. So remember, euchromatin, U is true chromatin, and it is more diffuse and not as compact. And so the genes are accessible for all the enzymes. Another thing that happens before the RNA is made is that eukaryotes have transcription factors. Those are really important. And then as the RNA is made, it's processed. So you've got the capping and the tailing, you've got splicing. All those things happen before it leaves and exits out through nuclear pore. So it gets transported to the cytoplasm. And then the ribosome, there's some regulation there. We didn't talk about too much about the regulation of the ribosome, except for the fact that it needs the 5' cap is more important than the tail, but I think both of them are important. And then what do we talk about proteins? Proteins get glycosylated. Sometimes they get modified in other ways. And we talked about these signal peptide things. I want to talk about that for a minute. So I want to just go through this diagram here one more time. So it says here, proteins that need to go to the ER have a signal peptide. Okay, so what is a signal peptide? A signal peptide is the first few amino acids. It's about 20 at the beginning of a gene. So this is kind of what it looks like. The signal peptide is a bunch of amino acids there. It usually has a pretty big hydrophobic section, and there's usually a lysine in there at the beginning. And this is a signal. It's a chemical signature that says to the cell, hey, this protein, it needs to be brought to the rough endoplasmic reticulum. So that starts to get made, and you can see there's our little protein. It's at the ribosome, and it's not even being done yet. And so notice that there's the signal peptide, and there it is. And there's also this other thing, the signal recognition particle. So the signal recognition particle, this is made of protein and RNA, and it's going to bind that signal. So it's going to have a hydrophobic pocket. It's going to grab that thing, and it's going to bring it over to the rough endoplasmic reticulum. So make sure you know definitions of these things and what they do. Okay, I'm not going to ask you to write a long answer question on this concept, but definitely some shorter answer or multiple choice style questions. So remember that there's RNA involved here, and this signal recognition particle RNA is an RNA type. And I'll come back to the RNA types in a moment. So here's the rest of the diagram. You can see it basically brings it over to the ER. It binds a receptor, and then translation continues, and the protein itself is made and inserted into the endoplasmic reticulum, and eventually gets delivered to another part of the cell after it goes through the Golgi body. So I've got a little video here, although I'm running out of time, but this is in the PowerPoint. I think it's pretty short, but if you want a different explanation of the signal recognition particle, take a look at the PowerPoint and play this YouTube video. So almost done. I got a couple more things to say to you. I might keep you 1 or 2 minutes over time here. So remember, there are the 6 types of RNA. So I want you to memorize this table here and be prepared to replicate it or do a matching question based on it. I've done both in the past, so you need to be able to do that and know all 6 types of these RNA for the final exam. So make sure you do that. That should be 6 marks if you can remember what all these types of RNA are. And now just a word to the wise. If I ask you what, let's say this one here. Let's say I ask you what type of RNA does this. Don't just put mRNA. Give me the full name. I want you to show me that you know the whole thing. Okay. Topic 19 was on recombinant DNA and technology. So make sure that you know a little bit about what each of these technologies can do. So basically a definition about what they can do. And make sure you go through some of the examples I shared in class as well, so that you get a little bit of an idea of some of the things out there that have been done. So we also have our forensics and paternity testing. I know I'm running out of time, so I'm just going to go through this super quick. Remember, we're looking for matching bands. So remember, the mom is going to have 50% of her bands match, and the true dad is going to have 50% of his bands match with the child, and there should be none with anybody who is a false father. So check, check, X. Take a look at this question. I'm not going to tell you the right answer right now, but this is a little bit more what the multiple choice question on the final exam is going to look like. So it's going to ask you, which of the following statements is consistent with the band, right? Or with the bands on the gel. There's a real gel, so I'm going to try to find a real gel for your exam. And usually they're relatively easy to interpret. There's some terms. So I want to finish off on a couple of things. I was kind of going through the topics and thinking about the final exam and thinking about the themes of Biology 107. So these are some of the things that we talked about this semester in terms of processes, right? So, go through and know a little bit about these kind of things, because I think all of these are important concepts around cell biology. So you should know what these things are, right? At a minimum, like I said, the earlier units, it's going to be, I'm not going to ask you long answer questions on photosynthesis or on exocytosis or anything like that, but you should know what those things are. In terms of major themes, I was thinking that these are the major themes of the course, right? Cell structures, cell processes, energy metabolism, the different types of cells, prokaryotes and eukaryotes, and of course, viruses that are not cells. And then the last part of the course is collectively called molecular biology. So while you're reviewing, just keep in mind, what these themes are. So that is the end. So that is the last lecture for Biology 107. Unless you attend the review session tomorrow. I want to wish everybody good luck on your final exams, and thank you for taking my course. I know it's not exactly the same doing everything online, but if you see me around the college or around town after this, say hi to me. Tell me you took my class. I may or may not recognize you because we did everything online, but let me know how it went and how you're doing. I'd love to hear from you anytime I see you or in the future. So best of luck on your exams and your future endeavors, and I hope to see you around someplace. Keep in mind, I will be available during the next few days for consultation for the final exam. So if you need to get a hold of me, please feel free to do so. Other than that, I hope you have a wonderful summer and take care.